The overall goal of this Drosophila larval sensory neuron injury model is to combine in vivo live imaging, two-photon laser axotomy dendriotomy, and the powerful fly genetic toolbox into a platform for screening potential promoters and inhibitors of neuroregeneration.
This method will help address key questions in the field of neuroregeneration, such as by identifying new intrinsic and extrinsic regulators for neuroregeneration, both in the peripheral and central nervous system.
The main advantage of this technique is that novel candidates for neuroregeneration can be screened in an easy, fast, and cheap way.
Though this system can provide insight into neuroregeneration, it can also be applied to other systems, such as neurodegenerative diseases and neuron-glia interactions.
Generally, individuals new to this method will struggle because different experimental setups that utilize different types of neurons are used to model axon versus dendritic regeneration.
For the larvae collection, prepare culture bottles as follows. Use a blade to punch a 1.5 centimeter hole in one wall of a Drosophila culture bottle and fill the hole with a cotton ball for ventilation. Then put a dab of yeast paste on a grape juice agar plate and use the plate to plug the main opening of the bottle. In such a bottle, set up a cross of 10 virgin females and 5 males and change the plate daily while culturing at 25 degrees Celsius. Culture the collected plates with a wet tissue soaked in mild propionic acid to prevent contamination. From the cultured plates, harvest larvae at the required stage using forceps. Gently transfer the